Deinonychus. Deinonychus/Deinonychus/Deinonychus, from Greek, delta epsilon iota nu sigma dienos, terrible and nu upsilon xionox, genitive nu upsilon chi omicron sigma onachos claw, is a genus of dromaeosaurid theropod dinosaur with one described species, Deinonychus anteropus. This species, which could grow up to 3.4 meters, 11 feet, long, lived during the early Cretaceous period, about 115 to 108 million years ago, from the mid-Aptian to early Albian stages. Fossils have been recovered from the U.S. states of Montana, Utah, Wyoming, and Oklahoma, in rocks of the Cloverleaf Formation, Cedar Mountain Formation, and Antlers Formation, though teeth that may belong to Deinonychus have been found much farther east in Maryland. Paleontologist John Ostrom's study of Deinonychus in the late 1960s revolutionized the way scientists thought about dinosaurs, leading to the dinosaur renaissance and igniting the debate on whether dinosaurs were warm-blooded or cold-blooded. Before this, the popular conception of dinosaurs had been one of plodding, reptilian giants. Ostrom noted the small body, sleek, horizontal posture, ratite-like spine, and especially the enlarged raptorial claws on the feet, which suggested an active, agile predator. Terrible claw refers to the unusually large, sickle-shaped talon on the second toe of each hind foot. The fossil YPM 5205 preserves a large, strongly curved ungual. In life, archosaurs have a horny sheath over this bone, which extends the length. Ostrom looked at crocodile and bird claws and reconstructed the claw for YPM 5205 as over 120 mm, 4.7 in, long. The species name Anteropus means counterbalance, which refers to Ostrom's idea about the function of the tail. As in other dromaeosaurids, the tail vertebrae have a series of ossified tendons and super-elongated bone processes. These features seem to make the tail into a stiff counterbalance, but a fossil of the very closely related Velociraptor mongoliensis, IGM 100-986, has an articulated tail skeleton that is curved laterally in a long S shape. This suggests that, in life, the tail could bend to the sides with a high degree of flexibility. In both the Cloverleaf and Antlers formations, Deinonychus remains have been found closely associated with those of the ornithopod Tenontosaurus. Teeth discovered associated with Tenontosaurus specimens imply they were hunted, or at least scavenged upon, by Deinonychus. Discovery and naming. Fossilized remains of Deinonychus have been recovered from the Cloverleaf Formation of Montana and Wyoming and in the roughly contemporary Antlers Formation of Oklahoma, in North America. The Cloverleaf Formation has been dated to the late Aptian through early Albian stages of the early Cretaceous, about 115 to 108 Ma. Additionally, teeth found in the Arundel clay facies, mid-Aptian, of the Potomac Formation on the Atlantic coastal plain of Maryland may be assigned to the genus. The first remains were uncovered in 1931 in southern Montana near the town of Billings. The team leader, paleontologist Barnum Brown, was primarily concerned with excavating and preparing the remains of the ornithopod dinosaur Tenontosaurus, but in his field report from the dig site to the American Museum of Natural History, he reported the discovery of a small carnivorous dinosaur close to a Tenontosaurus skeleton, but encased in lime difficult to prepare. He informally called the animal Daptosaurus agilis and made preparations for describing it and having the skeleton, specimen AMNH 3015, put on display, but never finished this work. Brown brought back from the Cloverleaf Formation the skeleton of a smaller theropod with seemingly oversized teeth that he informally named Megadontosaurus. John Ostrom, reviewing this material decades later, realized that the teeth came from Deinonychus, but the skeleton came from a completely different animal. He named this skeleton Microvenator. A little more than 30 years later, in August 1964, paleontologist John Ostrom led an expedition from Yale's Peabody Museum of Natural History which discovered more skeletal material near Bridger. Expeditions during the following 2 summers uncovered more than 1,000 bones, among which were at least 3 individuals. Since the association between the various recovered bones was weak, making the exact number of individual animals represented impossible to determine properly, the type specimen, YPM 5205, of Deinonychus was restricted to the complete left foot and partial right foot that definitely belonged to the same individual. The remaining specimens were catalogued in 50 separate entries at Yale's Peabody Museum, although they could have been from as few as 3 individuals. Later study by Ostrom and Grantee Meyer analyzed their own material as well as Brown's Daptosaurus in detail and found them to be the same species. Ostrom first published his findings in February 1969, giving all the referred remains the new name of Deinonychus anteropus. The specific name Anteropus, from Greek nu tau rho rho omicron pi omicron sigma, means counterbalancing and refers to the likely purpose of a stiffened tail. In July 1969, Ostrom published a very extensive monograph on Deinonychus. Though a myriad of bones was available by 1969, many important ones were missing or hard to interpret. There were few postorbital skull elements, no femurs, no sacrum, no furcula or sternum, missing vertebrae, and, Ostrom thought, only a tiny fragment of a coracoid. Ostrom's skeletal reconstruction of Deinonychus included a very unusual pelvic bone, a pubis that was trapezoidal and flat, unlike that of other theropods, but which was the same length as the ischium and which was found right next to it. Further findings. In 1974, Ostrom published another monograph on the shoulder of Deinonychus in which he realized that the pubis that he had described was actually a coracoid, a shoulder element. In that same year, another specimen of Deinonychus, MCZ 4371, was discovered and excavated in Montana by Stephen Orszag during a Harvard University expedition headed by Farish Jenkins. This discovery added several new elements, well-preserved femora, pubes, a sacrum, and better ilia, as well as elements of the pes and metatarsus. Ostrom described this specimen and revised his skeletal restoration of Deinonychus. This time it showed the very long pubis, and Ostrom began to suspect that they may have even been a little retroverted like those of birds. A skeleton of Deinonychus, including bones from the original, and most complete, AMNH 3015 specimen, can be seen on display at the American Museum of Natural History, with another specimen, MCZ 4371, on display at the Museum of Comparative Zoology at Harvard University. The American Museum and Harvard specimens are from a different locality than the Yale specimens. Even these two skeletal mounts are lacking elements, including the sterna, sternal ribs, furcula, and gastralia. Even after all Ostrom's work, several small blocks of lime-encased material remained unprepared in storage at the American Museum. These consisted mostly of isolated bones and bone fragments, including the original matrix, or surrounding rock in which the specimens were initially buried. An examination of these unprepared blocks by Gerald Grellet-Dinner and Peter Makovicky in 2000 revealed an interesting, overlooked feature. Several long, thin bones identified on the blocks as ossified tendons, structures that helped stiffen the tail of Deinonychus, turned out to actually represent gastralia, abdominal ribs. More significantly, a large number of previously unnoticed fossilized eggshells were discovered in the rock matrix that had surrounded the original Deinonychus specimen. In a subsequent, more detailed report, on the eggshells, Grellet-Dinner and Makovicky concluded that the egg almost certainly belonged to Deinonychus, representing the first dromaeosaurid egg to be identified. Moreover, the external surface of one eggshell was found in close contact with the gastralia, suggesting that Deinonychus might have brooded its eggs. This implies that Deinonychus used body heat transfer as a mechanism for egg incubation, and indicates an endothermy similar to modern birds. Further study by Gregory Erickson and colleagues finds that this individual was 13 or 14 years old at death and its growth had plateaued. Unlike other theropods in their study of specimens found associated with eggs or nests, it had finished growing at the time of its death. Implications. Ostrom's description of Deinonychus in 1969 has been described as the most important single discovery of dinosaur paleontology in the mid-20th century. The discovery of this clearly active, agile predator did much to change the scientific, and popular, conception of dinosaurs and open the door to speculation that some dinosaurs may have been warm-blooded. This development has been termed the dinosaur renaissance. Several years later, Ostrom noted similarities between the forefeet of Deinonychus and that of birds, an observation which led him to revive the hypothesis that birds are descended from dinosaurs. 40 years later, this idea is almost universally accepted. Because of its extremely bird-like anatomy and close relationship to other dromaeosaurids, paleontologists hypothesize that Deinonychus was probably covered in feathers. Clear fossil evidence of modern avian-style feathers exists for several related dromaeosaurids, including Velociraptor and Microraptor, though no direct evidence is yet known for Deinonychus itself. When conducting studies of such areas as the range of motion in the forelimbs, paleontologists like Phil Senner have taken the likely presence of wing feathers, as present in all known dromaeosaurs with skin impressions, into consideration. Description. Based on the few fully mature specimens, Deinonychus could reach 3.4 meters, 11 feet, in length, with a skull length of 410 millimeters, 16 in, a hip height of 0.87 meters, 2.9 feet, and a weight of 73 kilograms, 161 pounds, though there is a higher estimate of 100 kilograms, 220 pounds. The skull was equipped with powerful jaws lined with around 70 curved, blade-like teeth. Studies of the skull have progressed a great deal over the decades. Ostrom reconstructed the partial, imperfectly preserved skulls that he had as triangular, broad, and fairly similar to Allosaurus. Additional Deinonychus skull material and closely related species found with good three-dimensional preservation show that the palate was more vaulted than Ostrom thought, making the snout far narrower, while the juggles flared broadly, giving greater stereoscopic vision. The skull of Deinonychus was different from that of Velociraptor, however, in that it had a more robust skull roof, like that of Dromaeosaurus, and did not have the depressed nasals of Velociraptor. Both the skull and the lower jaw had fenestrae, skull openings, which reduced the weight of the skull. In Deinonychus, the antorbital fenestra, a skull opening between the eye and nostril, was particularly large. Deinonychus possessed large hands, manus, with 3 claws on each forelimb. The first digit was shortest and the second was longest. Each hind foot bore a sickle-shaped claw on the second digit, which was probably used during predation. No skin impressions have ever been found in association with fossils of Deinonychus. Nonetheless, the evidence suggests that the Dromaeosauridae, including Deinonychus, had feathers. The genus Microraptor is both older geologically and more primitive phylogenetically than Deinonychus, and within the same family. Multiple fossils of Microraptor preserve pennaceous, veined feathers like those of modern birds on the arms, legs, and tail, along with covert and contour feathers. Velociraptor is geologically younger than Deinonychus, but even more closely related. A specimen of Velociraptor has been found with quill knobs on the ulna. Quill knobs are where the follicular ligaments attached, and are a direct indicator of feathers of modern aspect. Classification: Deinonychus anteropus is one of the best-known dromaeosaurid species, and also a close relative of the smaller Velociraptor, which is found in younger, late Cretaceous age rock formations in Central Asia. The clade they form is called Velociraptorini. The subfamily name Velociraptorini was first coined by Rynch and Barsbold in 1983 and originally contained the single genus Velociraptor. Later, Phil Currie included most of the dromaeosaurids. 2 late Cretaceous genera, Zyagin from Mongolia and the North American Sauronatholestes, may also be close relatives, but the latter is poorly known and hard to classify. Velociraptor and its allies are regarded as using their claws more than their skulls as killing tools, as opposed to dromaeosaurines like Dromaeosaurus, which have stockier skulls. Phylogenetically, the dromaeosaurids represent one of the non-avialan dinosaur groups most closely related to birds. The cladogram below follows a 2015 analysis by paleontologists Robert De Palma, David Burnham, Larry Martin, Peter Larson, and Robert Backer, using updated data from the Theropod Working Group. This study currently classifies Deinonychus as a member of the Dromaeosauridae. Paleobiology. Predatory behavior. Deinonychus teeth found in association with fossils of the ornithopod dinosaur Tenontosaurus are quite common in the Cloverleaf Formation. 2 quarries have been discovered that preserve fairly complete Deinonychus fossils near Tenontosaurus fossils. The first, the Yale Quarry in the Cloverleaf of Montana, includes numerous teeth, 4 adult Deinonychus and 1 juvenile Deinonychus. The association of this number of Deinonychus skeletons in a single quarry suggests that Deinonychus may have fed on that animal, and perhaps hunted it. Ostrom and Maxwell have even used this information to speculate that Deinonychus might have lived and hunted in packs. The second such quarry is from the Antlers Formation of Oklahoma. The site contains 6 partial skeletons of Tenontosaurus of various sizes, along with 1 partial skeleton and many teeth of Deinonychus. One Tenontosaur humerus even bears what might be Deinonychus tooth marks. Brinkman et al. 1998, point out that Deinonychus had an adult mass of 70 to 100 kilograms, whereas adult Tenontosaurs were 1 to 4 metric tons. A solitary Deinonychus could not kill an adult Tenontosaur, suggesting that pack hunting is possible. A 2007 study by Roach and Brinkman has called into question the cooperative pack hunting behavior of Deinonychus, based on what is known of modern carnivore hunting and the taphonomy of Tenontosaur sites. Modern archosaurs, birds and crocodiles, and Komodo dragons typically display a little cooperative hunting. Instead, they are usually either solitary hunters, or are drawn to previously killed carcasses, where much conflict occurs between individuals of the same species. For example, in situations where groups of Komodo dragons are eating together, the largest individuals eat first and will attack smaller Komodos that attempt to feed. If the smaller animal is killed, it is cannibalized. When this information is applied to the Tenontosaur sites, it appears that what is found is consistent with Deinonychus having a Komodo or crocodile-like feeding strategy. Deinonychus skeletal remains found at these sites are from subadults, with missing parts consistent with having been eaten by other Deinonychus. On the other hand, a paper by Lee et al. Describes track sites with similar foot spacing and parallel trackways, implying gregarious packing behavior instead of uncoordinated feeding behavior. Contrary to the claim crocodilians do not hunt cooperatively, they have actually been observed to hunt cooperatively, meaning that the notion of infighting, competition for food and cannibalism ruling out cooperative feeding may actually be a false dichotomy. In 2009, Manning and colleagues interpreted dromaeosaur claw tips as functioning as a puncture and gripping element, whereas the expanded rear portion of the claw transferred load stress through the structure. They argue that the anatomy, form, and function of the foot's recurved digit 2 and hand claws of dromaeosaurs support a prey capture/grappling/climbing function. The team also suggests that a ratchet-like locking ligament might have provided an energy-efficient way for dromaeosaurs to hook their recurved digit 2 claw into prey. Shifting body weight locked the claws passively, allowing their jaws to dispatch prey. They conclude that the enhanced climbing abilities of dromaeosaur dinosaurs supported a scansorial, climbing, phase in the evolution of flight. In 2011, Denver Fowler and colleagues suggested a new method by which Deinonychus and other dromaeosaurs may have captured and restrained prey. This model, known as the Raptor Prey Restrained (RPR) model of predation, proposes that Deinonychus killed its prey in a manner very similar to extant accipitrid birds of prey, by leaping onto its quarry, pinning it under its body weight, and gripping it tightly with the large, sickle-shaped claws. Like accipitrids, the dromaeosaur would then begin to feed on the animal while still alive, until it eventually died from blood loss and organ failure. This proposal is based primarily on comparisons between the morphology and proportions of the feet and legs of dromaeosaurs to several groups of extant birds of prey with known predatory behaviors. Fowler found that the feet and legs of dromaeosaurs most closely resemble those of eagles and hawks, especially in terms of having an enlarged second claw and a similar range of grasping motion. However, the short metatarsus and foot strength would have been more similar to that of owls. The RPR method of predation would be consistent with other aspects of Deinonychus's anatomy, such as their unusual jaw and arm morphology. The arms were likely covered in long feathers, and may have been used as flapping stabilizers for balance while atop struggling prey, along with the stiff counterbalancing tail. Its jaws, thought to have had a comparatively weak bite force, might be used for saw motion bites, like the modern Komodo dragon, which also has a weak bite force, to finish off its prey if its kicks were not powerful enough. Bite force. Bite force estimates for Deinonychus were first produced in 2005, based on reconstructed jaw musculature. This study concluded that Deinonychus likely had a maximum bite force only 15% that of the modern American alligator. A 2010 study by Paul Gignac and colleagues attempted to estimate the bite force based directly on newly discovered Deinonychus tooth puncture marks in the bones of a Tyrannosaurus. These puncture marks came from a large individual, and provided the first evidence that large Deinonychus could bite through bone. Using the tooth marks, Gignac's team were able to determine that the bite force of Deinonychus was significantly higher than earlier studies had estimated by biomechanical studies alone. They found the bite force of Deinonychus to be between 4,100 and 8,200 newtons, greater than living carnivorous mammals including the hyena, and equivalent to a similarly sized alligator. Gignac and colleagues also noted, however, that bone puncture marks from Deinonychus are relatively rare, and unlike larger theropods with many known puncture marks like Tyrannosaurus, Deinonychus probably did not frequently bite through or eat bone. Instead, they probably used their high bite force in defense or prey capture, rather than feeding. Limb function. Despite being the most distinctive feature of Deinonychus, the shape and curvature of the sickle claw varies between specimens. The type specimen described by Ostrom in 1969 has a strongly curved sickle claw, while a newer specimen described in 1976 had a claw with much weaker curvature, more similar in profile with the normal claws on the remaining toes. Ostrom suggested that this difference in the size and shape of the sickle claws could be due to individual, sexual, or age-related variation, but admitted he could not be sure. There is anatomical and trackway evidence that this talon was held up off the ground while the dinosaur walked on the third and fourth toes. Ostrom suggested that Deinonychus could kick with the sickle claw to cut and slash at its prey. Some researchers even suggested that the talon was used to disembowel large ceratopsian dinosaurs. Other studies have suggested that the sickle claws were not used to slash but rather to deliver small stabs to the victim. In 2005, Manning and colleagues ran tests on a robotic replica that precisely matched the anatomy of Deinonychus and Velociraptor, and used hydraulic rams to make the robot strike a pig carcass. In these tests, The talons made only shallow punctures and could not cut or slash. The authors suggested that the talons would have been more effective in climbing than in dealing killing blows. In 2009, Manning and colleagues undertook additional analysis dromeus or claw function, using a numerical modeling approach to generate a 3D finite element stress/strain map of a Velociraptor hand claw. They went on to quantitatively evaluate the mechanical behavior of dromeus or claws and their function. They state that dromeus or claws were well adapted for climbing as they were resistant to forces acting in a single, longitudinal, plane, due to gravity. Ostrom compared Deinonychus to the ostrich and cassowary. He noted that the bird species can inflict serious injury with a large claw on a second toe. The cassowary has claws up to 125 mm, 4.9 in, long. Ostrom cited Gilliard, 1958, in saying that they can sever an arm or disembowel a man. Coffrin, 1999 and 2003, studied 241 documented cassowary attacks and found that 1 human and 2 dogs had been killed, but no evidence that cassowaries can disembowel or dismember other animals. Cassowaries use their claws to defend themselves, to attack threatening animals, and in agonistic displays such as the boat threat display. The sirima also has an enlarged second toe claw, and uses it to tear apart small prey items for swallowing. In 2011, a study suggested that the sickle claw would likely have been used to pin down prey while biting it, rather than as a slashing weapon. Biomechanical studies by Ken Carpenter in 2002 confirmed that the most likely function of the forelimbs in predation was grasping, as their great lengths would have permitted longer reach than for most other theropods. The rather large and elongated coracoid, indicating powerful muscles in the forelimbs, further strengthen this interpretation. Carpenter's biomechanical studies using bone casts also showed that Deinonychus could not fold its arms against its body like a bird, avian folding, contrary to what was inferred from the earlier 1985 descriptions by Jacques Gaudier and Gregory S. Paul in 1988. Studies by Phil Center in 2006 indicated that Deinonychus forelimbs could be used not only for grasping, but also for clutching objects towards the chest. If Deinonychus had feathered fingers and wings, the feathers would have limited the range of motion of the forelimbs to some degree. For example, when Deinonychus extended its arm forward, the palm of the hand automatically rotated to an upward-facing position. This would have caused one wing to block the other if both forelimbs were extended at the same time, leading Center to conclude that clutching objects to the chest would have only been accomplished with one arm at a time. The function of the fingers would also have been limited by feathers. For example, only the third digit of the hand could have been employed in activities such as probing crevices for small prey items, and only in a position perpendicular to the main wing. Alan Gishlig, in a 2001 study of Deinonychus forelimb mechanics, found that even if large wing feathers were present, the grasping ability of the hand would not have been significantly hindered. Rather, grasping would have been accomplished perpendicular to the wing, and objects likely would have been held by both hands simultaneously in a bear hug fashion, findings which have been supported by the later forelimb studies by Carpenter and Center. In a 2001 study conducted by Bruce Rothschild and other paleontologists, 43 hand bones and 52 foot bones referred to Deinonychus were examined for signs of stress fracture. None were found. The second phalanx of the second toe in the specimen YPM 5205 has a healed fracture. Parsons and Parsons have shown that juvenile and subadult specimens of Deinonychus display some morphological differences from the adults. For instance, the arms of the younger specimens were proportionally longer than those of the adults, a possible indication of difference in behavior between young and adults. Another example of this could be the function of the pedal claws. Parsons and Parsons have suggested that the claw curvature, which Ostrom had already shown was different between specimens, maybe was greater for juvenile Deinonychus, as this could help it climb in trees, and that the claws became straighter as the animal became older and started to live solely on the ground. This was based on the hypothesis that some small dromaeosaurids used their pedal claws for climbing. Flight. In a 2015 paper, it was reported after further analysis of immature fossils that the open and mobile nature of the shoulder joint might have meant that young Deinonychus were capable of some form of flight. Speed. Dromaeosaurids, especially Deinonychus, are often depicted as unusually fast-running animals in the popular media, and Ostrom himself speculated that Deinonychus was fleet-footed in his original description. However, when first described, a complete leg of Deinonychus had not been found, and Ostrom's speculation about the length of the femur, upper leg bone, later proved to have been an overestimate. In a later study, Ostrom noted that the ratio of the femur to the tibia, lower leg bone, is not as important in determining speed as the relative length of the foot and lower leg. In modern fleet-footed birds, like the ostrich, the foot-tibia ratio is 0.95. In unusually fast-running dinosaurs, like Struthiomimus, the ratio is 0.68, but in Deinonychus the ratio is 0.48. Ostrom stated that the only reasonable conclusion is that Deinonychus, while far from slow-moving, was not particularly fast compared to other dinosaurs, and certainly not as fast as modern flightless birds. The low foot-to-lower leg ratio in Deinonychus is due partly to an unusually short metatarsus, upper foot bones. The ratio is actually larger in smaller individuals than in larger ones. Ostrom suggested that the short metatarsus may be related to the function of the sickle claw, and used the fact that it appears to get shorter as individuals aged as support for this. He interpreted all these features, the short second toe with enlarged claw, short metatarsus, etc. as support for the use of the hind leg as an offensive weapon, where the sickle claw would strike downwards and backwards, and the leg pulled back and down at the same time, slashing and tearing at the prey. Ostrom suggested that the short metatarsus reduced overall stress on the leg bones during such an attack, and interpreted the unusual arrangement of muscle attachments in the Deinonychus leg as support for his idea that a different set of muscles was used in the predatory stroke than in walking or running. Therefore, Ostrom concluded that the legs of Deinonychus represented a balance between running adaptations needed for an agile predator, and stress-reducing features to compensate for its unique foot weapon. In his 1981 study of Canadian dinosaur footprints, Richard Cool produced rough walking speed estimates based on several trackways made by different species in the Gathing Formation of British Columbia. Cool estimated one of these trackways, representing the ichneus species Arenachnites gracilis, which may have been made by Deinonychus, to have a walking speed of 10.1 km per hour, 6 miles per hour. Eggs. The identification, in 2000, of a probable Deinonychus egg associated with one of the original specimens allowed comparison with other theropod dinosaurs in terms of egg structure, nesting, and reproduction. In their 2006 examination of the specimen, Grella-Tinner and Makovicky examined the possibility that the dromaeosaurid had been feeding on the egg, or that the egg fragments had been associated with the Deinonychus skeleton by coincidence. They dismissed the idea that the egg had been a meal for the theropod, noting that the fragments were sandwiched between the belly ribs and forelimb bones, making it impossible that they represented contents of the animal's stomach. In addition, the manner in which the egg had been crushed and fragmented indicated that it had been intact at the time of burial, and was broken by the fossilization process. The idea that the egg was randomly associated with the dinosaur was also found to be unlikely. The bones surrounding the egg had not been scattered or disarticulated, but remained fairly intact relative to their positions in life, indicating that the area around and including the egg was not disturbed during preservation. The fact that these bones were belly ribs, gastralia, which are very rarely found articulated, supported this interpretation. All the evidence, according to Grella-Tinner and Makovicky, indicates that the egg was intact beneath the body of the Deinonychus when it was buried. It is possible that this represents brooding or nesting behavior in Deinonychus similar to that seen in the related troodontids and oviraptorids, or that the egg was in fact inside the oviduct when the animal died. Examination of the Deinonychus egg's microstructure confirms that it belonged to a theropod, since it shares characteristics with other known theropod eggs and shows dissimilarities with ornithischian and sauropod eggs. Compared to other maniraptor and theropods, the egg of Deinonychus is more similar to those of oviraptorids than to those of troodontids, despite studies that show the latter are more closely related to dromaeosaurids like Deinonychus. While the egg was too badly crushed to accurately determine its size, Grella-Tinner and Makovicky estimated a diameter of about 7 cm, 2.7 in, based on the width of the pelvic canal through which the egg had to have passed. This size is similar to the 7.2 cm diameter of the largest Cytipodi, an oviraptorid. Eggs, Cytipodi and Deinonychus also shared the same overall body size, supporting this estimate. Additionally, the thicknesses of Cytipodi and Deinonychus eggshells are almost identical, and since shell thickness correlates with egg volume, this further supports the idea that the eggs of these two animals were about the same size. A study published in November 2018 by Norell, Yang and Wieman et al. Indicates that Deinonychus laid blue eggs, likely to camouflage them as well as creating open nests. The study also indicates that Deinonychus and other dinosaurs that created open nests likely represent an origin of color in modern bird eggs as an adaptation both for recognition and camouflage against predators. Life cycle. A study on Deinonychus tooth isotopes suggests precociality in the genus. The isotopes examined for different aged specimens indicates that adults and juveniles had different diets across the various age groups. The examinations also indicate a lack of complex, cooperative social behavior that is generally seen in pack hunting animals, suggesting that Deinonychus had a more typical reptilian set of life stages. Paleoecology. Geological evidence suggests that Deinonychus inhabited a floodplain or swamp-like habitat. The paleo-environment of both the Upper Cloverleaf Formation and the Antlers Formation, in which remains of Deinonychus have been found, consisted of tropical or subtropical forests, deltas and lagoons, not unlike today's Louisiana. Other animals Deinonychus shared its world with include herbivorous dinosaurs such as the armored sauropelta and the ornithopods Cephalosaurus and Tenontosaurus. In Oklahoma, the ecosystem of Deinonychus also included the large theropod Acrocanthosaurus, the huge sauropod Sauroposeidon, the crocodilians Goniophilus and Polyxysuchus, and the gar Lepisosteus. If the teeth found in Maryland are those of Deinonychus, then its neighbors would include Astrodon and a nodosaur, called Priconodon, only known from teeth. The middle portion of the Cloverleaf Formation ranges in age from 115 plus or minus 10 ma near the base to 108.5 plus or minus 0.2 ma near the top. Cultural significance. Deinonychus were featured prominently in the novels Jurassic Park and The Lost World by Michael Crichton and the film adaptations, directed by Steven Spielberg. However, Crichton ultimately chose to use the name Velociraptor for these dinosaurs, rather than Deinonychus. Crichton had met with John Ostrom several times during the writing process to discuss details of the possible range of behaviors and life appearance of Deinonychus. Crichton at one point apologetically told Ostrom that he had decided to use the name Velociraptor in place of Deinonychus for his book, because he felt the former name was more dramatic. Despite this, according to Ostrom, Crichton stated that the Velociraptor of the novel was based on Deinonychus in almost every detail, and that only the name had been changed. The Jurassic Park filmmakers followed suit, designing the film's models based almost entirely on Deinonychus instead of the actual Velociraptor, and they reportedly requested all of Ostrom's published papers on Deinonychus during production. As a result, they portrayed the film's dinosaurs with the size, proportions, and snout shape of Deinonychus.